welcome to our department colloquium series. Very delighted to welcome our speaker who had to take several flights of stairs to come to the seminar today. He's one of our own, Keith Brown. He's a postdoctoral fellow with the International Institute for Nanotechnology. And Keith is a physicist, trained on some obscure school on the East Coast. He got his undergraduate degree in physics from MIT and a graduate Ph.D. degree at Harvard. Then he came to Northwestern. For the last several years, he has been
at least he finished with a good one.
Yeah. Then he wisened up and came to the Midwest, Northwestern. The last couple of years or so, he's been working at the interface between chemistry, materials, physics, and biology using scan probe microscopy. Even at this early tenure, he has almost 3 dozen archival papers, number of awards for young researcher with ABS and IIN, and also defense graduate program. And his talk today will be about pushing the limits and making the room at the bottom is scanning probe microscopy. So let's welcome Keith Brown.
All right, well, thank you very much for the invitation to be here. I've had the pleasure of coming to these IIN Frontiers in Nanotechnology seminars for a couple of years, and I'm really honored to be a part of this tradition. So thank you very much for this invitation. Today, I'd like to tell you about some of the research I've been doing, both here at Northwestern, but also previously in my graduate work, all related to the theme of making room at the bottom with scanning probes. Now, many of you probably recognize that's an homage to the famous lecture delivered by Richard Feynman, There's Plenty of Room at the Bottom, in which he challenged us, scientists and engineers alike, to think about the benefits and opportunities that we could get by miniaturizing, by thinking at the very small scales. Well, needless to say, he was extremely correct. You only need to look as far as integrated circuits and microelectronics to see areas where miniaturization has revolutionized our lives. You know, this is the technology that's enabled things like your smartphone or the internet. And it all started only a few years after this famous lecture. In 1962, the first integrated circuit was developed with 16 transistors. And now you can buy from Intel in nearly the same area more than 1.5 billion transistors. So this miniaturization has really been this dominating factor. But I want to emphasize that this ability to pattern these hard materials on the small scales isn't just a technological achievement. It's also enabled an enormous amount of new science. In particular, you can think of at least 4 Nobel Prizes in physics that were given to basically achievements based upon the idea of patterning hard materials at the nanoscale. For example, the charge couple device or the observation of giant magnetoresistance. So it's not an exaggeration to say that a lot of what we know about semiconductor physics is based upon our ability to pattern these kinds of hard materials. So this is really transformative advanced scientifically. Unfortunately, this toolbox that we have is not generalizable. In particular, if you were to buy a circuit or a chip about 10 years ago, it would only have 14 elements in it in total. Moreover, and so that while the situation's improved in the following years, it's still very limiting as to what you can include in these processing. Furthermore, this whole idea of having sacrificial materials and then high energy beams and sequential liftoff processing, this is not generalizable to the soft materials that are important in many areas of study, in particular soft electronics and also moving into bioengineering. So we need better tools. And if you think about this, many of the interesting advances in nanoscience in the past couple of decades have come from things you might consider to be beyond the conventional toolbox of this nanofabrication. For example, think about semiconductor quantum dots, little crystals of semiconductor that have amazing luminescent properties, have been a toolbox and a playground for semiconductor physics, and also have been remarkably useful for imaging applications in biology. These were not made by some top-down process. These were made by a wet chemical process. Furthermore, think about graphene, this amazing material which has spawned a whole new field of study of two-dimensional electronics, two-dimensional materials. This is not isolated with some high-powered microscope, but it was isolated first with Scotch tape. So really, these are coming from, the point I'm trying to make with these examples is that some of these advances are coming from really different places from what we're used to thinking about. And so as a result, for people that are interested in nanoscience, I think it's important to ask ourselves, where will the next scientifically important nanostructures be coming from? And what will they look like? And I would say that many of the areas that exist now in study need advances in nanopatterning. For example, think about heterogeneous catalysis. It's well established at this point that small metal nanoparticles are very good at catalyzing chemical reactions due to their high surface area to volume ratio. Also, they can be made with enormous variety in their shape, and their size, and their composition. And so this is a great boon. Unfortunately, understanding the structure-function relationships that govern these transformations is very difficult. And this is because of the scale of these experiments. And so what we'd like to be able to do but can't is actually synthesize massive arrays of nanoparticles in which we know the composition and size of every nanoparticle in this array, so we can study in a very combinatorial level these kind of catalytic transformations. But we can't create things like this yet. You can also think about biomolecular arrays. So the Genome Project and the genetic revolution is powered by technologies like the ability to pattern biomolecules on a very small scale, which allow you to introduce other biomolecules like DNA and be able to sequence these at high speeds. And so part of this, we're experiencing this Moore's law-like decrease in cost in these kinds of technologies now, which have amazing payoffs in other fields. But how do we keep this going? And part of this is being able to pattern soft materials on smaller and smaller scales. Furthermore, as we continue to make these smaller and smaller, what happens now when these biomaterials become in the same scale as what cells expect to see in their native environment? Can we start to use these to template the behavior of cells on these scales? And then finally, if we could synthesize, most of this work is done with DNA, but if we could start to synthesize other more complicated molecules and constructs like proteins, enzymes, and sugars, all with these same kind of ease of use, what are the sort of implications for the other kinds of omics that you're used to thinking about and hearing about? And so the point that I really like to make with these examples is there are plenty of examples of hard and soft materials that we'd like to be able to pattern better at the small scale. And they're huge scientific and technological opportunities for doing this. So if you're interested in this challenge and you're saying, OK, how can we learn about better how to pattern things on small scales? Well, one way that I like to think about it personally, I have a very kind of reductionist point of view, take a complicated process and try to boil it down to the simplest parts and build from there. So one thing you could say is, well, how do we control and understand physical and chemical processes on very small scales? So one way you could think about doing this is imagining just a point in space interacting with a surface. And can we tune and observe this kind of interaction? And so, of course, this is a very abstract concept. We can't do this exactly. But what we can do is use modern instrumentation, for example, the scanning probe or atomic force microscope, that features a very sharp tip at the end of a flexible cantilever. And we can move this whole construct in 3 dimensions with nanoscale resolution. And we can use this sensitive cantilever to measure and apply piconewton scale forces. And this has been a remarkable tool for nanotechnology because it directly gives you access to the nanoscale world. And so what I'd like to tell you about, I should mention this has been going on, research in this area has been going on for about 30 years now, with a huge variety of applications in both imaging and manipulation. And so what I'd like to tell you about today are 3 examples from my own research of basically fundamental studies of tip sample interactions that lead us to the ability to create new kinds of nanoscale architectures. And I'll tell you about 3 examples from my research. The first is from my PhD research, really motivated by looking at fields like optical tweezers, where people have been using non-contact trapping of particles as a way of both assembling structures and also studying biochemical and biophysical interactions. And they've learned a lot from this. And we'll ask the question, is it possible to do this with a scanning force probe? And what can we learn by doing this? The second area I'll talk about is introduce you, and for many of you, reintroduce you to dip-pen nanolithography, or the ability to directly pattern material from a tip to a surface, and talk about how this has been widely accepted as a diffusion-based process, and how we've been able to show recently that it's actually in many cases better thought of as a fluid flow, and tell you about the implications of that. And then finally, I'll talk about a big overarching question of throughput. And so many of the scanning probe studies really focus on extremely small areas, and this is due to the architecture on which they're based. And so I'll introduce you to a Cantilever-free architecture and tell you about how we've been able to show that this has a lot of potential for overcoming some of these limitations. And then I'll end by telling you about some of the implications and future directions of this work. So to begin, I'll address the question, is it possible to hold a particle without touching it at the end of a scanning probe? OK, but returning to the sort of central theme I've set up here is, how can we learn how to position particles on small scales better? Right? And so you might think, OK, if you're kind of construct some nanoscale system, the simplest way to do it, perhaps, is to synthesize the pieces and put them together like you would something in the macro scale. And there's a lot of good reasons to think about things this way. First of all, we're really good at making pieces. Right? So here's an example, from our research in the Mirkin group. We're able to synthesize metal nanoparticles with very high control over their size and shape. And so maybe it's possible to be able to actually position these at the single particle level and build up complicated structures. And so this is an appealing idea. And so people have thought about this, of course. And one of the earliest ways people have thought about doing this is a technology they call microgrippers. Perhaps we can just miniaturize tweezers down to the nanoscale. And this technology is basically based on either two scanning probes or other kind of MEMS concepts. And what people find is you can do this. You can use this to pick up sort of microscale objects and move them and drop them much like you would tweezers. The problem is as you scale this down and try to pick up smaller and smaller objects, the van der Waals forces that you can't turn off become more important than the body forces that you can. And so what ends up happening is you can't let them go. And so this is known in the literature as the sticky finger problem. And so one way to get over this is, okay, well, let's just avoid touching things. And so people, of course, have worked out ways to do this, the most popular of which is certainly optical tweezers, where by shining light to a fixed point in space, you can create a high potential there for particles to be trapped through a combination of the gradient force and the optical pressure. And as such, you can actually hold particles without touching them. You can use this for either biophysics, as I've mentioned, or for assembly. And people have explored a multitude of kinds of schemes here. And the problem with this, though, is that the wave nature of light limits you. And so since white light has, in the visible range, has a few hundred nanometer wavelengths, this means that you can only focus this down so much. And no matter how much you turn up the trapping potential, you're going to have only limited control over the position of this object, and ends up limiting you in nanopositioning on the order of a few hundred nanometers. And so the question that we asked as part of my graduate research is it possible to combine this non-contact trapping of things like optical tweezers with the ability and the physical precision of things like micro grippers? And so as a way to think about how to start this, we need a way to turn on and off an interaction to start with. And so we thought about, at first, electrical fields. So electrical fields are great because you can turn them on and off very easily. So if you have a particle in an electrical field, it's situations like this. If the particle is not charged, or if you simply use an AC field that's going back and forth very quickly, what you find is there's no electrophoretic motion, but you in fact polarize the particle, generating a net dipole. Now if the field is uniform, this is the end of the story. However, if the field is non-uniform, you experience a situation where the particle is actually pulled to the maximum electric field using an effect known as dielectrophoresis. And so this is an appealing approach because it's insensitive to things like the charge. So perhaps this is useful. So is one way to think about this. We can say, okay, well, here we have a scanning probe tip. The simulation is done with this tip being charged at 1 volt and the ground down here being held at 0, at grounded. And what you see basically from this picture is that this is a very colorful picture. And that's actually a bad thing. So I'm showing you the potential here. And the fact that it's colorful means that the potential, and thus the electric field, is changing in all of space. Therefore, this interaction is not very localized to the tip. So this is not a very good way of trying to localize this interaction. And so as a way to try to overcome this, we developed a new kind of scanning probe tip, where we actually can try to localize electrostatic interaction at the very tip. So in particular, we developed this new kind of probe where we have a conductive shell that goes all the way down to the very tip. We call these coaxial probes because of their similarity to coaxial cables. So you can apply a voltage at this tip with respect to these biased shells here. And thus, you can create a very localized interaction. So we can actually create these. This was one such probe made at the clean room at Harvard, where we have this inner conductor here and an outer shell. And so perhaps we can use these kinds of probes for this kind of assembly I've talked about. And so a typical experiment might proceed as follows. So we have a surface. We've decorated it with a series of randomly dispersed micron scale beads. And we come in with this probe. Remember, it's an AFM, so we can use this to image. And so we can use this probe to try to find the particles of interest. So for example, we perform a tapping mode image, and we see this sort of arrangement of strange triangular-looking things. And so these, they're actually 1 micron diameter beads. But the reason they look sort of triangular is because this tip itself is sort of triangular. And so what you see is the convolution between the tip and the particles of interest. So they look this shape. But nonetheless, we can use this as a way to localize these and try to pick them up. So we can energize the tip, press it against this bead of interest, and withdraw it. Once we do that, we find that we actually, so one thing we see is that, first of all, they're no longer triangular. Now all these particles appear spherical. And this is because now they're being imaged not by this triangular tip, but by the bead that's trapped at the tip of this probe. But also we notice that the bead we attempted to grab is missing. So we're able to grab this as we predicted. And we can put it down in a desired location. So this is demonstrating that we can do this kind of micron scale assembly with this approach. However, if we try to scale this down like I anticipated, I said we would need to, what we find is as we go to the nanoscale particles, we can no longer let them go. This sticky finger problem rears its ugly head. However, this time thinking about these kinds of probes is not wasted. The fact that we create these really highly concentrated electric fields ends up being very useful for imaging. In particular, we have a very highly localized field right at the tip of this probe. And so we thought about a couple different ways this could be useful for imaging surfaces. It's a bit of a tangent. So for example, we found that using this highly localized force, we can actually image the topography of the surface rather than using this force for manipulating materials. We can use this for imaging them, and we can get high-resolution images of materials we otherwise couldn't see with these probes. Additionally, we can use these for electrostatic force microscopy and try to basically read material properties. In this case, we're looking at the work function or contact potential. And we're able to show about an order of magnitude improvement over probes that are not coated. So these have potential for imaging. But getting back to the main storyline, I told you about using these for manipulation of materials, try to position objects. And so the situation is actually quite bad, because if you look at the electrostatic limit, it's actually not possible to have a field maxima that's not touching an electrode. So this interaction, as I've described it, cannot be used to do this kind of non-contact manipulation. Fortunately, this is a little bit more subtle than I've led you to believe. In particular, this is not dependent just on, this can be a medium that's non-trivial. In particular, it can be dependent upon the dielectric permittivity of the particle and the medium. As such, the situation described, if the particle is more polarizable than the medium, then the particle is attracted to the maximum of the field as I've described. However, if the particle is less polarizable than the medium, you get a different situation where the medium is pulled towards the tip and the particle is actually pushed away. So to think about why this might happen, think about, gravity is always an attractive force in sort of daily life situations, but helium balloons rise. And this is because the air is pulled down more strongly. And so that's the perfect analogy to why this could be used for both positive and negative interactions here. And so the probe I've described to you, this coaxial probe, creates a field maximum. So this is actually pretty bad for this kind of study because, of course, the positive DV particles are pulled in, the negative ones are just pushed away, and that's kind of the end of the story. So we propose to use a slightly more complicated trapping probe where we actually have 3 independently controllable electrodes, where we have this core electrode as we had before, and then 2 shell electrodes, an inner and outer shell. And by applying a potential on just the inner shell electrode, we find that we can actually create a 0 in the electric field displaced from the tip. And so this is, we call this a triaxial probe. It's kind of an electrical engineering term because triaxial cables have 3 coaxial shells as this one does. And so we can make these, and presumably the particle will be held in this trap. Again, we can make these. This is another probe that I made in the clean room at Harvard. We have the inner core electrode and then 2 shells surrounding it. And so the next thing I'll show you is a video of trapping particles with this probe. And it'll be in this section here, and it'll be top down, the same perspective you're looking at here, and the particles will be below and the tips up here. The thing I want to draw your attention to, though, is the fact that these parameters can depend on things like frequency. So we can actually, by just changing the excitation, we can change whether this is an attractive or repulsive interaction. And that's important because that means we can draw particles towards the tip with one frequency and then flip the polarization and trap them. So what I'll show you basically, and I'll start it now, is a movie of this is the tip, and we're drawing particles towards it, and then we can flip the interaction, ejecting the particles that are not in the trapping region, and then you see some particles that are sort of held in this trap. I'll start this again. So you see here we are drawing particles towards the tip, and we flip the interaction. So these are fluorescent particles, which allows us to visualize them. They're polystyrene particles in a water glycerol mixture. So we can repeat this process till we have a single particle held in this trap. And you see here is a movie of such a situation. And we can collect a lot of this information and do statistics on this to try to understand the trapping potential. And we do this, we can calculate based on the principal component analysis of where this particle is. We can find the trapping axes, and we can decompose this into different these major and minor axes. And we can use this to say, okay, well, how well are we holding this particle? And so this is a 200 nanometer diameter polystyrene particle. So we're holding it in a region commensurate with its size. So that's good. But also, we can think about what I think is a bit more interesting is the shape of this trap. And the reason I think the shape is very interesting is we can compare that very easily to the shape predicted by a model, for example. So if we construct a simple model of the situation, forget about the whole 3-dimensional structure of this, the fact that it looks somewhat triangular, but replace all of that with just a point electrode and 2 rings that are capacitively coupled, and use this as a way to try to calculate what these forces should be. What we find is that the shape, based upon the ratio of the 2 trapping widths, ends up matching very, very closely with what we see in experiment, telling us that we have a very good picture of what's going on in this situation. And so in this section, I really told you about some of the advantages of thinking about how we could craft electric fields at the tips of these probes, and how that could be very useful for not only imaging very fine features and new kinds of properties, but also for holding particles without trapping them. And we initially got interested in this because we wanted to construct quantum mechanical systems where we position single particles next to one another and such. But what we realized as we were going through with this is that the particles held in these traps could themselves be useful as a probe, because we have the ability to hold them and manipulate them. And so in thinking about that, we wanted to compare this and benchmark this against some of the literature in the biophysics community, where people think about doing this kind of force spectroscopy, where there you consider parameterizing your system based upon things like the stiffness of your trap, the size of the probe you can accommodate. And there the typical techniques are like optical tweezers, magnetic tweezers, and AFM. And what we find is we can use the model that I showed you earlier, this ring model, as a way of calculating, in the best case, what this kind of mode can do. And what we can see is we can generate sort of a allowed region of trapping as a function of particle radius and the size of the tip, basically, that gives us where this would sit. And we find in the sort of highest terms is that we can potentially trap particles much smaller than can be trapped through things like optical tweezers, but with force constants very, very similar, or even better in many cases. So this has a lot of potential, I believe, in that kind of force spectroscopy biophysics areas. Okay, so from here, sort of in my career, I graduated with a Ph.D., and I was very interested in continuing this line of reasoning, but it became clear that if you're trying to create structures with these kind of scanning probes, if you're doing this in a single particle at a time scale, this is going to be prohibitively slow. And if you ever want to make larger scale structures or do larger scale experiments, this isn't going to be effective. So that drew me to working with Professor Mirkin, who has been pioneering this technique, dip-pen nanolithography, which is a direct write way of patterning materials, which in principle can be a whole lot faster and more useful. And so in DPN, this is a technique that uses a scanning probe, as I've talked about before, but it takes advantage of something that has been a problem in the field of scanning probe imaging for a long time. And that is that if you take a scanning probe and touch it on the surface in ambient conditions, then capillary condensation will actually make a meniscus form that in the context of imaging is very bad, because it means you have this extra force term that convolutes what you'd like to be looking at. But in DPN, this is extremely useful, because what you can do is you can coat the tip with some material of interest. Here, pictured here are some alkane thiol molecules. And you can use this meniscus as a way to transport them from the tip onto the surface. And the initial experiments, they found that you can actually pattern self-assembled monolayers of these molecules on gold structures. And so I think this was very appealing to researchers for two reasons. One is that this is a direct write technique that allows you to pattern a lot of different kinds of things, including biomolecules, proteins and DNA, for example, things that are very difficult to pattern by any other means. And also, this is a very interesting physical problem, because there's a lot going on, yet it can be thought about somewhat simply. And in fact, the Schatz group came up with a very nice model very early on based upon diffusion from the tip onto the surface, in which they found that if you replace all of this complexity with just a point source and you deliver material at a constant rate, so they called this the diffusion model because that's what diffusion would do in this case, what you find is that you can actually match the data with small molecules very well. And the situations you find the volume of the feature grows linearly with time. And so this was very appealing, but in the years that followed, people sort of moved a bit away from the small molecule materials and into larger molecules, things like agarose and polyethylene glycol. And the reason that these are interesting is because these can act as essentially resins that transport other materials that you could have difficulty patterning otherwise, so biomaterials or other nanomaterials, whatever you might be interested in. And so you could really be patterned in a really general way. And so this, this is a technique that we've been using for this kind of study for many years now. But people still think about this in the context of this diffusion-based model. But if you look at this more carefully, there's a lot of conflicting evidence. For example, if the features you write with these kinds of probes, they're not flat anymore. They're dome-like, which suggests that perhaps surface tension is playing a role in the patterns that are generated. Furthermore, when we do studies and we use inks that have higher viscosity, we find that transport is slower, something you might expect, but also suggests that perhaps there's a fluidic component to this. Despite all of this, if you read through the DPN literature, what you'll find is that people will always plot their data as feature size versus time to the 1/2, which bears with it all of these assumptions of this small molecule diffusion-based system that goes back many years. And so this is something we wanted to address. In particular, we wanted to ask, what can we learn by thinking about these not as a diffusion process, but one as a fluid flow? So in order to study this, we constructed a model system where we patterned inks composed of polyethylene glycol. So we take a tip, we coat it with this material, we hold it in contact with the surface, we hold it for some time, and we withdraw the tip. And what we find is that there's a polymer feature left behind. And we can characterize the size of that feature with atomic force microscopy, for example, calculating its volume. And the first thing we studied was the dwell time, because this is the property that people have been using to characterize the size of features basically the whole time in the DPN community. And so what we find, for example, if we pattern onto a hydrophobic surface as fluorosilane-treated silicon, we can look at the volume deposited as a function of dwell time. And we see, first of all, it's very small. We're patterning things smaller than an attoliter. So these are smaller than 100 nanometers on a side. And you can see this is increasing extremely gently with dwell time. Here, the power law slope is only 0.15. So it's basically almost independent of dwell time. And so what's a bit more interesting, though, is if we pattern on a hydrophilic surface, the situation is a bit more complicated. And we see that, first of all, the features are much larger, but also it increases much more rapidly to the 0.5 power approximately. And so this is an interesting number, because for those of you that you've taught or studied fluids know that if you have capillary invasion or wicking processes, typically scale as volume goes as time to the 1/2. So this is suggesting perhaps this could be related to some kind of wicking process. But additionally, neither of these power laws are 1. So this is telling that this other foundational model maybe doesn't apply in these cases. Maybe this really is treating these like a fluid flow is more appropriate. And so if that's the case, then the second part of this process that's completely been ignored, this process of capillary rupture, maybe this matters a lot. And so we wanted to study this process. And so in particular, we wrote a series of features with the same dwell time, but changing the speed at which we withdrew the tip. And what we found is we patterned on hydrophilic silicon, so untreated silicon. We found that the features, as we slowed the process down, they got bigger. This was not a surprise. This basically told us is that as we hold the tip in contact, patterning is happening forward in time. That was great. However, if we pattern on the hydrophobic silicon, what we found is as we withdrew the tip more slowly, the features got smaller, which puzzled us for a while until I dug up a paper from the Soviet Journal of Colloid Physics from 1983, which showed a series of experiments where basically the experimenters, they took a drop of fluid, put it between two plates, and then withdrew the plates at different speeds. And what they found is if you draw them very fast, then the identity of the plates in the fluid doesn't matter. The droplet is split in half. But as you slow this down, you basically give the fluid time to decide which plate it wants better, and it moves to the surface with the lower contact angle. And so this mirrors qualitatively what we see. We see that as you slow this process down, you're giving the fluid a chance to realize that it would rather not be on the surface and would rather be on the tip. We can be a bit more quantitative about this and treat this as more of a fluids problem and try to construct a number that would characterize the competition between something like surface tension and net viscosity, and that is a capillary number. And when we do this, we find that this transition is associated with a capillary number on the order of 10 to the minus 5, which if you compare this to literature with what people see in sort of nanoscale flows is in very good agreement. And so this really shows you that here this process with these kinds of inks really is better thought of as a fluid flow. And that means a different series of parameters are important. So first of all, the surface energy is very important, as I've showed. But also, the withdrawal speed plays a big role. And that realization actually ends up being quite important, because what it means is if you withdraw the tip very slowly, maybe you can make very small features. And indeed, what we found is if we withdrew the tip at a rate of 100 nanometers per second, we can make features down to 14 nanometers in size. And so these features, these are made up of polymers that have 2,000 molecular weights. This is actually only about 40 polymer molecules in this case. So these are about the smallest. This is certainly the smallest that I've found of any polymer feature that's been directly written. And it's right up at the edge of the smallest DPN feature that's ever been written by any means. So this is really telling you that this realization of going slowly with the withdrawal speed has a lot of implications. And so you might ask, okay, so we're patterning polyethylene glycol. It's a pretty simple material. Is this useful for anything? And I would say yes. There's actually, we needn't choose such a simple material. In particular, a lot of the work in the Mirkin group that's been going on in parallel with this has to do with developing a new technique called scanning probe block copolymer lithography, where you're not patterning a simple polymer. Instead, what you're patterning is a block copolymer, where you have one block that's the same material, polyethylene glycol, which is mediating the transport, but another block, polyvinylpyridine, which is actually coordinating to metal ions in solution. And what this allows you to do is to pattern not just a simple polymer material, but that with the associated network of ions. And then if you treat this at high temperature, it's actually possible to anneal these into a single or coalesce these into a single particle that we can then remove the polymer with further treatment. And so what that allows us to do basically is to synthesize on surfaces wherever we decide to place them a series of nanoparticles. And we've shown compositions such as gold, silver, platinum, palladium, and also oxides and alloys are possible. Alloys, of course, we can just change the ratio of the precursor materials. And so I want to bring this back to what I told you about earlier about how we'd like to study things like catalysis, and how now by thinking about these, this DPN process as a fluid flow, we now have an avenue to patterning extremely small metal particles, the size of which could be very useful for doing these kinds of studies. Okay, so the final area I'd like to tell you about is really comes down to a question of throughput, and that's that really all of the examples I've told you about thus far, and really if you've seen many scanning probe work before, it's all a very small area, sort of tens of microns on a side at most. And this is because of the nature of the work. You're scanning at a very nanoscale tip. You're scanning slowly across a surface. And so this is a big problem. If you want to be able to address large area samples, if you want to address the area of many cells on a surface or something like this, or a wafer if you're thinking about electronics, you need to overcome this. And so people have thought about using many huge arrays of many cantilevers to do this. And a lot of work has gone into this. Unfortunately, basically the problem with this work is that when you start to do this, the arrays themselves go from many tens of dollars or something for a single cantilever to thousands of dollars for big arrays, at least. And so as a result, you just don't do the experiments. If you think about a grad student working in a lab trying to do an experiment where you mess up and drop it, you're out a few $1,000s. That's going to limit the amount of times you're willing to try that experiment. And that's just in practice what's happened. These technologies have been around for over a decade. Just they haven't been applied. And so what's needed is an architectural innovation. And this is really what the innovation that drew me to the Mirkin group back when I joined a few years back. And in particular, what they were able to show is that you don't need cantilevers. What they were able to show is that instead of using a cantilever, you can use simply a film of a soft material, in this case elastomer, and then you won't support a single tip. You can support a huge array of tips. And then this all has to be on a transparent backing layer to provide this whole thing in a flat plane and make it transparent so you can see through what you're doing. And the nice thing about this kind of approach is you can think about this being made very, very simply, right? So you have these arrays of elastomeric probes. This whole part of the structure can mold in one step. And in fact, they are able to show in this first demonstration, you can make as many as 11 million pens on a 3-inch wafer in one step using materials that are extremely cheap, thus sort of cutting this cost barrier away from this process. So in the years that followed this initial demonstration, they were able to basically demonstrate this could be used for lithography and some of the unique aspects of that. But the thing that, when I first joined that I think was very interesting is the sort of culture was saying, okay, we have these probes, we've lost a lot by going away from these cantilever-based systems. How can we get it back, right? How do we get back towards the performance we had with DPN? And I took a bit of an alternate approach by saying, okay, well, we actually have, a few decades or decade and a half of experience working with probes that are silicon or silicon nitride. What's different about using a probe that's an elastomer, that's a soft material? What's different about this? And what's unique? And so started looking into this and found a couple of very fascinating things. The first has to do with the permeability of soft materials. So the fact that if you have a soft material, it's possible, rather than these hard materials, you can actually absorb solvent from the ambient conditions, or you can deliberately put solvent in there. And what that means is that in DPN, you're reliant on this capillary condensation effect. What we're able to show is that using these probes, you can actually store water in these elastomeric materials in a way of patterning in totally dry environments without the need for environmental humidity at all. And what's, I think, a bit more enabling, though, is you can load these with any material you want that's permeable. So for example, if you load it with indecane, a nonpolar solvent, you can actually pattern things that are not soluble in water. So you're actually expanding the versatility of this technique beyond what was possible with the hard tips. Another interesting question is, okay, say you want to change the mechanical properties of these probes. Well, if you have ever bought an AFM probe, you know that you have to sort of go to the website, pick out the right spring constant, and match it with the material properties of the system you're interested in. So how do we do this here? So rather than thinking about the geometry of the probe, which is what you tune in sort of cantilever geometry, we can think about tuning the chemistry of these probes. And by adjusting, for example, the base to crosslinker density of these elastomers, we can actually change the mechanical properties over a very significant range. And we can change it from a range that's amenable to kind of nano-indenting experiments all the way to one that's in the intermediate of what people use scanning probes for in general, which is that of frequency modulation. And so I think what's a bit an interesting note about this as well is the fact that if you go to these websites and buy cantilevers, you'll notice that the spring constants are spec'd over an order of magnitude at least. And that's because when you make these probes, they're extremely sensitive to small deviations in their properties, because the things like the thickness of the cantilever, the length of the cantilever, they enter into the spring constant equation to the third power. And as such, small variations are amplified. Here in these systems, everything's linear. So we actually, in principle, have a better ability to specify properties of these probes at the outset. Another area we wanted to address was that these probes are all insulating, electrically insulating. But a lot of what people use these for are these kinds of probes is delivering electrical energy. So is it possible to render these conductive? And so one way we thought about doing this, well, these are large flat structures. Perhaps we can just coat them in a material like graphene. And we found that their architecture, the sort of flat extended network, allows you to do this. But one thing that this study led us to understand was that if we coat a tip with graphene, we can actually study, well, maybe that has something to do with that changes its mechanical properties, changes the interface between this probe and the surface. We performed a series of friction force microscopy experiments and found that in this case, when we coat these probes with graphene, we can actually reduce the tip sample friction by about 40%, improving their wear performance by many factors. And this is important because other people, this was demonstrated a couple of years ago, and since then, 2 other groups have shown that this is also the case. And so this really shows that people are thinking about wear in these scanning probe experiments, not just in the cantilever-free community, but really in the whole the whole scanning probe community. So this actually could be a pretty important observation. But I want to get back to what was the central challenge that was posed to me when I joined the group, and that was really summed up by this picture. So this is 20 copies of the Beijing Olympic logo. It's taken from a field of about 15,000 that were written in one experiment. And the problem is really illustrated by this, because this is, in this experiment, while you have this large array of probes, and while you can move them independently, so you can control what they are exactly, they're all mechanically held to one another. So whatever pattern you write with one is being written with all of them. And so the question is, well, how do we write arbitrary patterns? How do we stitch together patterns to make this actually a truly arbitrary pattern generating system? So one way you could think about doing this, or the challenge another way, is how do we take an array of probes that are fixed to one another and move one relative to the others? And this needs to be a scalable thing, right? Because the whole reason we moved to the system is that we wanted to get away from the sort of high-cost system. So how do we do this in a way that preserves this low-cost nature? And so looking at the material properties of this and thinking about how we could do this, I came up with the idea, well, okay, so this is made out of effectively a synthetic rubber, So one thing about rubbers is they have a really high coefficient of thermal expansion, right? And that could be a big problem in some cases. But here, maybe that's an advantage. Perhaps if we can put small heaters beneath each of these, we can actually locally heat these and move one probe relative to its neighbors. So in order to test this, we performed a series of experiments where we took basically a glass slide and we fabricated on top of this an array of coils that are indium tin oxide heater coils. They operate just like if you have an electric range. And by locally heating them, we can basically deliver heat to a specific coil. And we coat this with a layer of an elastomer, a very thin rubber. In this case, it was 90 microns. And we can observe the motion of this using a scanning probe microscope, an AFM. And what we found is if we delivered 40 milliwatts to a specific pen, we can turn it on and off, and we can observe a series of cooling and heating curves, as you see here. And when we do this, we see that we can actuate amplitudes of this specific location over 4 microns. And so this is an interesting number because, the sort of micron scale is what we need to actuate these. So this is certainly a large enough distance for kind of lithography. So that box is checked in some sense. But what's a bit more interesting, though, is the efficiency we get, sort of the distance per unit power we get in these systems is actually on par with what people get, because you can make bimorphic cantilevers that are hard materials, and you can use these as thermal actuators. And this is actually, this efficiency is on par with those systems. And this is because, in one way of thinking about it, we traded the mechanical advantage from the geometry for a materials advantage by using the soft material with this very high coefficient of thermal expansion. So I found that very fascinating. Additionally, these are moving very fast. So we're able to move these over 100 microns a second, in some cases up to about 1/3 of a micron per second. This is actually faster than we can move these with the piezo actuator in our lithography system. And so this is, good enough for lithography, certainly, but we wanted to understand a bit more about how this is working the way it does. And so we constructed a model to try to capture the phenomena using a simple linear heat diffusion model and linear actuation. And to give you a couple of summaries of this, so this would, for example, predict that the time it would take to be actuated would have to do with the amount of time it takes heat to diffuse from the heater into the elastomeric film. And indeed, we find that matches very well with our experiments. So it's really just based upon the thickness of the film and the thermal diffusivity of the material. This actually matches so well that we've been using this recently not just as a way to characterize the actuator, but when we construct composite films, films that are not, that don't have coefficients of thermal expansion we can look up in a textbook, we can actually use this as a way to measure these coefficients. And so this shows very good agreement with this model. But so taken together, what that really says is that we understand this property. Perhaps we can use this for lithography. So we should try to put that to the test. And so we constructed an array of 4x4 heater coils that we can independently address. On top of each of these coils, each of these black squares is actually one of these PDMS elastomeric pens. And we're going to try to use this for a lithography experiment. And so to do this, we're going to coat this with an ink using a small molecule in alkane thiol mercaptahexadecanoic acid. And we're going to try to write this onto a gold-coated silicon wafer. And a typical experiment, one where we sort of test the actuation, would look as follows. When we look at the region according to one pen that we're writing, we can try to write a series of features where we vary the power from the bottom left here going to 0 milliwatts all the way to 32 milliwatts in the top right. And what we find is that the features we write, first of all, we write features, so that's of course good. But they start out very small and round, and they get larger till they're large and square. And this matches our expectation of this system. When we're first in contact, we're based on diffusion, where the material is diffusing out from the spot. And as we push harder, we're deforming the tip, and we see the sort of cross-section of the pen. So with this in hand, we can try to write a large area and stitch together large patterns. And so here's a region of the periodic table that we try to write with 16 pens. And so I'll just briefly overlay the pen array that I showed you from the previous slide to give you a sense of scale. And so you can see that here, each pen is writing one element in its atomic number and mass. And so you can see that, doing this kind of experiment, we've now patterned the self-assembled monolayer on a gold surface, where we can control basically one molecule high surfaces across, in this case, about a millimeter. And so this is, showing you that we can actually affect this kind of independent addressability with these pen arrays. Now, kind of up to this point in the talk, I've really only discussed the idea of lithography as patterning materials, moving materials around, either picking and placing them or patterning them directly. But if you think about patterning more generally, the vast majority of what people do in the clean rooms or in companies has to do with directing energy. Light in particular is much more widely used. And so with this new architecture, we asked, is it possible to use this architecture to have some advantages in those regards, to be able to pattern things that other people can't do, or have some other kinds of advantages? And so one area you might want to pattern with high resolution is with light, because of its ubiquity and the wide range of photochemistries people have developed for that light can be used to modulate. And so one kind of experiment, well, I guess principally the challenge with light is the fact that it has, as I mentioned earlier, this finite wavelength, which means that if you try to pattern with high resolution, you have a diffraction limit where you just literally can't place 2 points closer together without playing some kind of trick. And this is because of the wave nature of light again. And so the trick that we can think about playing with these pen arrays is try to squeeze light through a small aperture, effectively operate in the near field. And so the way you can imagine doing this with these pen arrays, so these whole things are transparent, right? So, but if we try to coat these in an opaque material, for example, a thin layer of gold, we can perhaps leave a small aperture at the tip of each pen. If that aperture is sub-wavelength, maybe we can squeeze some light through it with the size commensurate with the hole, and we can affect some photochemistry below this pen, and then perhaps we can use some pattern transfer like deposition liftoff process to deposit some other kind of material if we choose to. And so if you want to pursue this, there's a couple of big challenges you have to think about. The first is, okay, well, what you're really asking is to make sub-wavelength apertures on a massive scale in some parallel process. And so this sounds very difficult. Additionally, you also have to figure out a way to get light to each of these pens independently, which again could be non-trivial. And so the way to think, I of course wouldn't be introducing this if we didn't find ways to address this. And unfortunately, I don't have time to go into the gritty details. But basically, the process by which we're able to make small apertures at the tip of each pen involves the fact that when you spin coat a material with a polymer, in a commonly used process in clean room fabrication kinds of techniques, when you spin coat a material that isn't flat, it's not going to coat it uniformly. So we can actually find ways to spin coat these pen arrays such that we can leave basically the tip of a given pen or all the pens sticking out just a little bit, and we can tune that by basically etching to correct for any sort of mistake. But the end result is by using this kind of batch technique, we're able to create pens that have 100 nanometer scale apertures with only about a 10% CV across the entire array. And we can characterize the amount of light that gets through these a bit better by doing experiments where we shine light through these and try to expose some photoresist. If we develop this, the photoresist gives us a kind of a physical imprint of where the light was. And we can do these kinds of experiments. We can try to write features very close together. We see that we can write features, this AFM trace shows that we can write features that are easily discernible when they're sort of 160 nanometer pitch. But more importantly, if you zoom in on these features and look at their full width half maxes, the distribution of the light is down at about 90 nanometers, which is in accordance with the size of the apertures we've made. So now, since we're using 405 nanometer light, we're clearly in the range where diffraction unlimited. We're working with the dominant length scale is not the wavelength of light, but the size of the aperture. And so the next challenge that we need to overcome is that of being able to get light to each pen independently. And so this could be difficult because of the fact that these apertures are very small, down to 100 nanometers. But you have to remember that these apertures are at the apex of pyramids that are on the 10 micron scale. So this is actually a large enough size scale that you can address these practically with far field optics. And so what we hypothesize is perhaps it's possible to use things like a digital micromirror device, the kind of instrument that's used even in the projector here perhaps, as a way of guiding light on these large scales. And so the basic concept was to couple this kind of digital micromirror device, which has as many as up to 800,000 pixels in this case, couple this with a pen array where we can actually direct light to each pen independently. And so this is a complicated problem that involves synchronizing a couple of disparate systems. And my contributions to this problem were really, a large part of them were trying to engineer the system that allowed this to take place in a reasonable time scale and with a high degree of automation. But I don't want to go into the details of that too much. But if you have questions, please ask, certainly. But basically, the end result is what we are able to do is control the position of this pen array and the light that gets to each pen and do all this independently. And so as a way to test this to see how well we can do, we can try to pattern over large areas. And so, for example, as a test of this, admittedly somewhat gimmicky one, we can try to write a map of the world. And so the pattern that we created is shown here. And so what you see, first of all, I guess this is a gold features that we've written on a silicon wafer using a liftoff process. And what you can kind of make out is the outline of continents, as you see here. The first thing I'll draw your attention to, though, is actually the scale bar. So this is a millimeter. This pattern is on the order of a centimeter across. So this was not taken with a microscope. This was taken with a camera. So this is actually the first time, to our knowledge, that any scanning probe technique has generated a pattern large enough that you can visualize with the naked eye. And so it's not only large, though, it's also intricate. If you zoom in on these kind of blob structures, you see that they're actually individual molecular formulae. So these are all taken from the PubChem database. This is Taxol, the cancer drug, for any biologists in the room get a kick out of that. But these are also made up of dot features. And there are also molecules and dot features in the ocean regions. And we've changed their size to give this contrast. In the ocean, they're down at about 150 nanometers. In the land, they're up at about 3/4 of a micron. And so if you're not a lithography aficionado, I can understand not knowing if this is to be impressed or not of this. So think about the following analogy. So if you take this pattern and blow it up to the size of the United States, then the individual dot features we write down here are the size of houses. So there's an extraordinary length scale between the smallest features we can write and the breadth of this pattern, which I think is really the key advance here. And so we wanted to prove the utility of this and do some sort of push the envelope and do things that are more functionally useful to people that are interested in nanotechnology. And one thing that we need to be able to do when creating a pattern like this is pattern with registry to existing structures, right? How do we actually put this where we want this on small scales? And one area where you need to do this is a really canonical problem that I'm sure many in this room are very familiar with, is how do we draw electrical contacts to nanostructures? So for example, if we have a nanowire, how do we draw wires to it? How do we solder it up? And so the way we proposed to do this was basically take this randomly dispersed silicon nanowires on a surface and basically take pictures of it with a high-resolution microscope, optical microscope, and then basically draw onto it what we wanted to print. And then we can take this and use this as the pattern we draw. So then we spin coat this with a photoresist, and then we expose this using our scanning probe technique. And you can see that the registration is quite good. And then we can do a deposition lift-off process to give us the hard electrodes that we need. And then I think the important thing about this is we actually perform measurements here to test whether or not this patterning worked. So here, for example, you can test this wire electrically and see this is in fact a functioning p-type transistor. And so this is the kind of experiment that many of you have done many times using techniques like electron beam lithography. And there you're sort of wiring up wires on the 1-by-1 basis. But in this experiment, we're actually wiring up 60 wires simultaneously across about a centimeter, or several millimeters anyway. And so you can see here a large area pattern showing you these wires. In this case, we were looking at 60 wires. After lithography, things looked great for 59 of them. And then when we did the testing, 2/3 of them worked. So this shows you that we have the ability to now construct this sort of functional architectures across very large scales. OK, so in the remaining few minutes, I'd just like to kind of summarize what I think are the important points of this and tell you about the directions this work is going forward. And really, at a very high level, what I've told you about is what I've learned from thinking about, just looking at tip sample interactions and what if you view these interactions very fundamentally, what we could learn about what are the new structures we could make. And how, if we think about crafting electromagnetic fields on the nanoscale, what that can give us in terms of being able to image with high resolution and hold particles without touching them. And then next, I introduced this technique of, or reintroduced, I suppose, dip and nanolithography, and how this can be used to deposit materials, and how, in many cases, this is actually really more of a fluid flow. And that simple realization gave us the ability to create extremely small features, smaller than had been patterned before of this kind of material, and how that advance can be used to do things like pattern small catalysts. And then I introduced this question of, is the cantilever necessary? And in fact, I hope I convinced you that not only is the cantilever not always necessary, but in fact, removing it, going away from this cantilever-based architecture, gives you a lot of advantages. Advantages in terms of scalability, of cost, and now I think also I've hoped to prove that in terms of versatility. I told you a lot about really, I guess taking a step back, if you could remember just sort of one picture to summarize this last section, it would be this one. And really what we're doing is trying to locally control properties from scales varying from the nanometer-scale to the centimeter-scale. And I've really focused on looking at illumination and controlling molecular species, but the same ideas can be used to control, and we've shown can be used to control things like pressure, electric potential, heat, and even the presence of solvent. And if you've been through a lot of sort of talks about how to create new nanostructures, I think an important thing to ask yourself at the end, an important thing to address is, well, where do we go from here? What are the unique kinds of structures that we're enabled, and what are the new scientific insights that these are going to provide? And so that's what I'd like to end by telling you about. And that's really actually the work that is now currently ongoing in the Mirkin group that these tools are enabling. And the first really comes back to the question that I posed early on, which is that of catalysis. So is it possible to synthesize on a very large scale arrays of nanoparticles that you can use to screen catalytic reactions? For example, can you create a large-scale, centimeter-scale array in which you have control over the size and composition such that you can do analysis of nanoparticles? And so this is work that we have ongoing. We're doing this in collaboration with GlaxoSmithKline, and I'm working with a pair of talented graduate students, including a materials science graduate student, PC over here, and also James. And they're doing some amazing work here with me. Additionally, another big area that is of interest is, I presented this idea of patterning materials and patterning energy as being disparate things. But one unique thing about this platform is this really gives you the ability to do both simultaneously. We can use the same pens to deposit materials and energy simultaneously if we prefer. And so one example that we've been able to show with this is being able to take these pens and deposit materials on a surface and then drive photochemistry with this same pen. In this particular case, we were depositing monomers that would form a carbohydrate. With this structure, then once we synthesize it, we can then use this to study the interactions between carbohydrates and proteins, which we're able to show. And these are very difficult structures to synthesize in general. And so this, from the perspective of the novel synthetic capabilities, this is quite enabling. And so I hope this is only the first of many examples of kinds of biochemistry experiments that one can perform using these kind of large-scale pen arrays. And this work is done in collaboration with a graduate student in chemistry, Shu He, and then the group of Adam Braunschweig in Miami. And so to conclude, I'd just like to thank the people that contributed to this work and helped me along the way. In particular, my Ph.D. advisor, Bob Westervelt. He's an amazing source of inspiration. He has a very clear way of looking at problems, and so he's been a great inspiration for me. My postdoctoral advisor, Professor Mirkin, has been great and very supportive, and also given me a lot of liberty to try things, make mistakes, and hopefully learn a lot. I'd also like to acknowledge the DipPen nanolithography subgroup with whom I've got many numerous collaborations. I also want to single out a few people that have moved on from the group. They're still alive. But Dan Eichelsdorfer, Xing Liao, and Weiyang Shim. This work was also funded by DARPA, FOSR, NSF, and I've been funded through the American Society for Engineering Education and the International Institute for Nanotechnology. So thank you for your attention. I'd be happy to answer any questions.
Oh.
So I think DipPen lithography has been in this regime of transferring inks through mechanical contact for a long time. Now that you have these kind of elastomeric tips where you can store ink inside the elastomeric tip, especially for combinatorial studies, is it possible to transfer and paint inks based on maybe pH, temperature, things that be important for biochemical, biophysical studies?
So you're proposing an experiment where you sort of template some, on the surface you template some variation where you have pH varying across the surface and you have sort of transport dictates, or this property dictates different kinds of transport across the array? Is this?
Yeah, if you're interested in mapping biochemical, biophysical properties based on local pH, but is it possible to do more printing, not just on looking and sticking the array where you want it to go on printing, but maybe looking at more complex properties dictating printing?
Yeah. I mean, I guess one thing that's really nice about these is that we know that their ability to pattern depends a lot on the molecular structure. So if you can envision many ways for molecular structure to change, right? You can go from inks that will print to inks that won't print very easily. And so you could have stimuli-responsive inks, for example, that would, in some cases, in some pHs or some light conditions or salt concentrations would pattern, and others they wouldn't. So that's a fascinating idea, and one we are actually sort of investigating some ideas of multi-stimuli responsive inks, currently.
How long does it take you to write that world map?
That world map took 8 hours to write, which is, we like that number because we benchmarked how long it would take to pattern that using the E-beam lithography system we have, and it was also 8 hours. So we could have raced, but that one took 8 hours. I will mention, though, that I don't bathroom break 8 hours? Okay. But in any case, the limitations, what's dictating that 8 hours in our case are nothing near fundamental. We're currently undergoing work. We have a talented research associate here who's helping us develop a way to basically up that by factors of a few thousand by doing simple things. Like in that experiment, we had we had a few hundred pens, each talking to or a few hundred mirrors on the DMD talking to a single pen, right? If you can make it such that you have a one-to-one registry between mirrors and pens, you get that factor already. So a factor in that case of 100. And then by upping the light source, which again is very possible, you can go a lot faster. So in this case, the fact that you have all these pens operating in parallel, I think the fundamental limit is very, very far away, and we're yet to reach it. But already we are able to reach what a $1 million system can do.
Sort of along those lines, what kind of software platform do you have for deconvoluting the image you want to draw into the series of pen movements? And is it optimized so that similar pen movements done by different pens can be done at the same time to reduce the drawing time?
Yeah, exactly. So this is a part of my contribution. I implemented a MATLAB script that basically takes the image and maps it into the different pens, and looks like you're kind of indicating. Looks for times when it can save time by not. So basically, when you're in a given location, it does all of the operations you need to do in that location, and it only goes to the locations that you need to go to, given the operations you need to write. So for the 1 million pen array, that actually doesn't really save you much time because you basically have to go everywhere. But for a 16 pen array or something like that, it cuts it way down, of course. But yeah, that was all done. That's a script all done in MATLAB.
Yes, Mark.
It seems like the fair comparison for the scanning near field optical microscopy would be immersion photolithography as opposed to e-beam lithography. And immersion photolithography is used by Intel, IBM, etc. can pattern 22 nanometer features at 12-inch wafer scale with 30 lithography steps and perfect registry.
Yeah.
So how would you compete against that technology?
My answer to that would be the fair comparison would not be against the near field immersion lithography, but against the mask. Because what if you don't need, I mean, I'm not saying that this technology is going to replace, what Intel is currently using on their fabrication line, right? I'm saying that, if you have an experiment that requires you to make a unique structure, maybe even informed by what you're looking at on the surface, that the ability to adapt to that and have basically a dynamic mask is very important in many cases. And the fact that it's, altogether much more inexpensive. So, if you make the comparison, yeah, of course, once you have the mask that you made with e-beam lithography tediously, and that mask is valued at many, millions of dollars or something like this. So that once you have that, you can crank those things out. But I think that's really, you can't ignore that piece as a generator of that structure.
The follow-up question is, when you have these pens, which are separate from one another, what is the precision in their respective positions? By that I mean, if you want to make a pattern which is not individual pixels, like your world map, each pixel is separate from one another. They don't have to connect to one another. How well can you control the connection point between overlapping pen patterns?
Yeah, so I didn't show it, but we performed a series of experiments where we patterned continuous lines and such. And we found that using the techniques that we currently have, I mean, the limitation certainly is when you do the optical lithography, it has to do with the way, how well the mask is made. And then, if there's any sort of residual heating and stress in the molding process. And so what we found is that in creating sort of 200 nanometer wide lines, that registry is good enough for that kind of capability. If you want to go beyond that, I imagine you'll have to do some kind of error correction, where you'll have to understand how they deviate from their ideal position and then correct that, which of course you can do by simply, if your resolution is, or your writing patterns are very 100 nanometers, you can just sort of shift where it's going to be. But yes, I mean, that's
you have to
certainly.
do that for each pen in the way, though.
Well, if that was the critical issue, you would eventually need to map that out. I mean, certainly, we know it's not as bad as 200 nanometers. We haven't studied, how much better it is than that. I think actually the non-uniform way the pens deform might be a bigger differentiator than just physically where they are. But yeah, that's something that we have not studied in detail beyond that. But I think if it's reasonable to think that it might be consistent the way it's off, especially if it has to do with the deviation in their original positions. In which case, if you measured it once, you could correct for it for a given pen array. But yeah, you have to measure for all the pens.
For each pen array.
For each pen array.
Let me ask one question as the DJ privilege. So most of your work is showing two-dimensional flat surfaces. What are the prospects to be able to do it on 3D surface? And there's one other question I think many would probably have liked to have asked. How did you find that Soviet journal reference?
So obviously Google Scholar is very useful, but I think the more useful thing is to dig, once you find a paper that seems relevant but doesn't quite have what you need, to dig backwards, right? And thankfully that paper was in English, right? It was an interlibrary loan from some other university I don't recall. Basically every time I give this talk, at least 2 people ask for it, so I'm happy to send it to you if you're interested. It's an old photocopy, certainly, but yeah. So, and your other question was about
going beyond 2D.
Right. So accommodating structures that aren't flat is that you or constructing structures in 3 dimensions.
So doing 3D nanostructuring or 3D fashion.
Can you turn it into a 3D printer?
Yeah. So our one is to be able to do it on a flexible substrate and then use the flexible substrate to create a 3D structure. Or would you be able to pattern across 3 dimensions?
Yeah. So we're actually investigating both of those directions. The easier one to think about is how do you pattern on things that aren't flat? And the answer there comes down to basically dictating the way the pens deform is the simplest way to think about that. I mean, so for example, if the pens are not soft materials, but if the pens themselves are hard, we actually have found that you can deform them to accommodate sort of many microns of deviation in the topography, and they'll accommodate that in their deformation. So in that scale, you can do that, right? The more challenging thing is patterning things in 3 dimension, because a lot of the tricks people use to do like a 3D printer, be it stereolithography or immersion printing or the sort of nozzle-based printing, they have problems when you get that small. be it surface tension, or if you're thinking about the stereolithography, you're trying to expose light, the very tip, you end up having the highest intensity of light right against the surface, right? So you might actually end up sticking the ink to the tip. So there's a lot of kind of practical challenges that I think there will be really interesting material solutions to them, but they're not straightforward. For example, you could imagine materials that rather than the materials themselves, just being like a simple stereolithography ink where you cross-link it where you shine the light, maybe it's actually bifunctional. When you shine one wavelength, you can cross-link some voxel, and then you shine a lower intensity of a different wavelength, and you decross-link or you sort of detach the tip from that. So it requires a little bit more materials innovation, but it's something that we're definitely looking into.
There are no more questions, so let's thank Keith for